Starch, protein and fat are all made up of large molecules. These food molecules must be broken down into smaller ones before they can be used by the body. Digestive enzymes speed up this process. Each enzyme breaks down a particular nutrient. Amylase breaks down starch. Proteases, like pepsin, break down proteins. And lipase breaks down fats. Each enzyme is released at a particular point in the digestive system. In the mouth, amylase is present in saliva. In the stomach, only pepsin is present. Most digestion takes place in the small intestine. pH varies in different parts of the digestive system. Does pH affect the way digestive enzymes work? To find out, first we'll investigate salivary amylase. There's an equal amount of starch solution in each of these 3 test tubes. A weak acid is added to the first. This solution is pH 3. A neutral solution is added to the second, pH 7. A weak alkali is added to the third, pH 9. Now, the same amount of salivary amylase is placed in each, and immediately a sample of each is tested with iodine to monitor the reaction. The iodine turns blue-black because starch is still present. Every 2 minutes, a drop from each test tube is added to the iodine. If the starch has been digested, the iodine will not change colour. Finally, 6 minutes into the experiment, it will work best. Here, salivary amylase works best at pH 7. This is its optimum pH. Next, we investigate pepsin, which is released in the stomach. There's protein, egg white solution, in these 3 test tubes. An acid is added to the first. A neutral solution is added to the second. An alkali is added to the third. The same amount of pepsin is added to each. As the egg white is digested, it'll change from a cloudy suspension to a clear solution. The solid particles of egg protein are broken down into smaller soluble particles. The test tube with acid is the only one which clears. In our experiment, the optimum pH for pepsin is pH 3. This is the pH in the stomach due to hydrochloric acid. Digestion continues in the small intestine, with enzymes released by the pancreas. One of these is lipase, needed to digest fats. But this process depends also on the presence of bile salts from the gallbladder. To show this, these 3 tubes contain some full-fat milk. A weak alkali is added, replicating the conditions in the small intestine. Next, we add an indicator, phenolphthalein. With the weak alkali, it's pink. Bile is added to test tubes 1 and 2. Then the same amount of lipase is added to test tubes 2 and 3. So, tube 1 contains bile salts. Tube 2 has both lipase and bile salts. Tube 3 has lipase. When fats are digested, they're broken down into glycerol and fatty acids. As the fatty acids neutralise the alkali, the pH level falls. This causes the indicator to lose its pink colour. Acidity increases most rapidly in tube 2, showing that fat is digested fastest in this environment. Tube 2 contains both lipase and bile salts, the optimum condition for digesting fat. As tube 1 shows, bile salts alone don't digest fat. Tube 3 shows that on its own, lipase is slow to digest fat. In tube 2, bile salts speed up lipase action by breaking up the fat into small droplets. This increases the surface area on which the lipase can act. Hungry for the nutrients in our food, our bodies are well tuned to create the optimum conditions for each enzyme to work most effectively.